Hi everyone, this is Tanisha Shekhawat, your zoology educator, and I welcome you all to PW Gulf. And today we have our revision session 1 of our evolution chapter. Okay, so evolution chapter we will be starting together. We will revise it properly. We also have to finish a slight bit left portion of the reproductive health that I also have today. Okay, so we'll be completing that first, and then we will move on to the evolution part. Let's wait for all the students to join first. Okay, so let's wait for 5 minutes. After that, we will be starting the session together. Hello, good evening, Jonas. Are you a new student? Good evening. Is your first time attending any of my classes? Hello, how are you? Okay, okay, got it, got it. Great then, great. So the thing is that obviously we were supposed to start a new chapter only today. We will revise evolution, but some portion of the previous chapter was left. Now the thing is only you are in class right now and you would not have much idea of the previous chapter, which is a reproductive health. So can you tell me, do you have any idea of reproductive health chapter? Have you ever studied that chapter before? Any idea about that particular chapter? It's reproductive health I am talking about right now. You can unmute yourself as well. Okay, okay, you got a little idea. Okay, then we will complete the art, that means assisted reproductive techniques portion was remaining from that chapter. I couldn't finish it in the previous session, but it is not a very huge portion. Hence, I brought it in today's chapter. Like I added a few slides of the previous chapter in front of evolution. So we will complete the ART, that means assisted reproductive techniques. After that, we will be completing the evolution. That means we will be starting the evolution. We will see how much we can cover. Okay, so let's start. First of all, we will be looking at what we have to cover today. So first thing that we have to cover is the introduction of the evolution chapter. Then we will look at the evolution. What exactly is evolution? After that, we will be completing the theories of evolution. That's it. Okay, moving forward. Now this is the reproductive health portion. I will see how to separate it, if it can be separated or something, but it is a revision, so I cannot just like put the entire class for a small portion of one particular chapter. Hence, I had to merge the chapters. Okay, so here we are talking about the transmission of STD. So this is from the NCERT. Because this is your first time attending class, I will tell you that I always try to carry NCERT along our sessions so that you read the important portions of NCERT in the class itself, and later on you do not have to like read it separately, because the NCERT language is very complex and it becomes very difficult to understand the language on your own without the help of a teacher. Okay, so this is the portion of STD, transmission of STD in your NCERT, and from this particular portion every year we see a lot of questions in your both boards and NEET. Okay, so if you are going for apart from NEET also, if you're going for some other competitive exam, there also this particular portion plays a very important role. So first of all, they will be talking about how hepatitis B and HIV, apart from sexual contact, can also be transmitted by blood contact. Okay, so blood contact means sharing of injection and needles, surgical instruments, and transfusion of the blood from infected person to a healthy person. Apart from that, from mother to fetus as well. Okay, so they're talking about which particular STDs, hepatitis B and HIV, both are viral caused STDs, sexually transmitted diseases, or we can say VD, venereal diseases, or reproductive tract infection, which is RTI. Okay, these three words can be used for STD. So no matter which particular word they are using, remember they are talking about STD only. So they are talking about how these two can be transmitted by these methods. Now, one very important question every time they ask is that which diseases among these are completely non-curable. Non-curable means once you get the infection, no matter which treatment, no matter what you try, you cannot completely cure from it. Okay, you can manage the symptoms, you can reduce the symptoms, but complete cure from these particular infections is definitely not possible. And if we talk about the HIV infection, it is a fatal, right? it can cause death of the person as well. So which are completely non-curable ones? Hepatitis B, genital herpes, and HIV infection. So this line is also very important, that these three are completely non-curable. Apart from that, if you detect any particular STD early on, okay, early on means timely, then they are completely curable. You can completely cure it. Okay, so how do we detect any STD, right, ma'am? We have to treat. So for that we have to basically diagnose ourselves early. So how do we perform diagnosis? Obviously tests are going to be there, but you will be going for those tests only and only if you see some particular symptoms of these STDs. So if we talk about symptoms, it can include itching, okay, fluid discharge, apart from that slight pain, swelling, etc. So this is the symptoms portion. This particular portion you have to highlight in your NCERT. They sometimes ask what is the symptoms of STD, early symptoms from which you can detect. But the problem with STDs is that they are usually very late in showing symptoms. Okay, early on detection of STD is very difficult because symptoms are very much delayed means they are seen very much later on. Once you get infection, like after months or years, you will be seeing the symptoms of that particular infection. Okay, so apart from that, infected females, these were the general ones which can be in males also and females also, these particular symptoms that we just highlighted. Okay, apart from that, specifically in females, what symptoms we can see? First of all, it is mostly asymptomatic. Especially in females, it is mostly asymptomatic. Hence, it becomes very difficult to diagnose and goes undetected for long. Apart from that, what problems it can cause? Okay, what problems it can cause? It can cause pelvic inflammatory disease. Pelvic inflammatory disease means the region, the groin region, the region where our reproductive organs and external genitalia is present, that is termed as a pelvis. Okay, infection of that pelvis will cause PIDs, pelvic inflammatory diseases. Okay, what happens if we get a PID? There will be abortions, stillbirth, ectopic pregnancy, and infertility, or even cancer is possible. Okay, so this particular portion that we have highlighted from this NCERT portion is extremely important. Okay, so this particular part is clear. If you have any doubts till now, you can ask. There isn't much, it is mostly theory, hence we usually cover it very, very quickly. Do you have any doubts, Jonas? Asymptomatic. Okay, great. See, if I have fever, like if I get an infection right now, though, so what can happen? Let's say I have a viral infection, rhinovirus, cold, which we commonly call. When virus enters my body, it is going to cause a lot of different changes in my body. It is going to impact a lot of different cells and machineries of my body. As a result, what will happen? As a result of those some changes in my body, I will see a few symptoms, such as I will see fever, runny nose, right, red eyes, tiredness, body pain. So all of this is a symptoms of the virus infection, right? Virus is harming our body from inside by taking over our machinery to reproduce itself. That is what virus is doing. On the other hand, everything that you see on outside is a side result of whatever virus has done to your body, which we from outside, if we see, we are going to call it a symptom, right? If I can check that I am having a fever right now, fever is a symptom. Now, asymptomatic would mean that you have the infection, but you are not showing any symptoms. Okay, from externally, you feel completely normal as a healthy individual only. But inside your body, the particular pathogen is reproducing and increasing its progeny, and it is slowly increasing the infection in the body on the inside, which later on, obviously, it is going to show up. It is going to cause a lot of issues to our body. But in the early phases, it was asymptomatic, means externally no impact was visible. Hence, we were not able to diagnose that whether the person is having any problem or not, right? We usually go to doctor. We usually, if we are healthy, we are not feeling any issue with ourselves, we do not go to a doctor to get some tests done. We only and only will go to a doctor when we see a symptom, right? Asymptomatic would mean that you are not seeing any symptom while you are having infection. Hence, infection keeps on increasing and the pathogen ultimately harms the body way too much. Clear? Great, great. Okay, we'll move forward now. Now, next up we have, so how can we avoid STDs, right? STD, a particular line has been written in our NCERT that STD are such a huge problem in our country and we need to somehow control it. How to control it then? Okay, avoid sex with multiple partners or unknown partners. Now, the thing is that the mode of transmission of majority of the STD is from a, one second, from a infected individual. If this is infected individual, from infected person, you will have transmission to a healthy person. Let's say this is a healthy person. Upon sexual contact, okay, if there is a sexual contact between these two, transmission will occur. So if we as healthy individuals make sure that healthy individuals are not having any kind of sexual contact with anyone who can be infected, right? How can we make sure that? Obviously, first thing, the other person with whom you're having any kind of sexual contact, you should know them well, right? You should know that they are absolutely not carrying any STDs. Second thing, multiple partner thing is that when you have multiple partners, one or the many partners from them can be carrying a STD, a particular pathogen which can be transmitted to you, right? And then you can also start transmitting to other people. So that is the concept of multiple partners. So you should completely avoid any kind of sexual contact with unknown partners which you don't know, you are not sure whether the person is having any STD or not, and multiple partners also, it should be avoided. Okay, second thing, always use condoms during coitus, right? When we study about the contraceptive methods, we know that condoms function as a great contraceptive method, right? That is there. So it is going to do one very good thing, very good thing, that it is a contraceptive method. Second thing, that apart from condoms, usually none of the other contraceptive methods that we have studied about, right, the IUDs or the barrier method of vault diaphragms or the oral pills, none of them completely avoids the sexual contact between the genitalia of the partners. Okay, on the other hand, condom completely creates a barrier between the genitalia of the partners if there is no sexual contact, because there is a barrier, the condom is present in between. If there is no sexual contact, obviously transmission cannot be there. Hence, only and only this particular, which is condoms, they function as a very good contraception as well as to avoid any kind of STD, a great measure to avoid STD as well. That is the second thing that you can do. Third thing, that obviously whenever you are in doubt, right, you might be seeing some symptom, you're not sure whether it is some STD symptom or not, but you still are slight doubtful. So you should quickly look for some healthcare professional and you should go for a consultation with them. Okay, so you should check out with the qualified doctors for early detection, because we know that if early detection is there, then it is completely manageable. Most of the STDs apart from hepatitis B, herpes and HIV are completely curable. So there is no issue. Okay, clear this part? Great, great, great. Now we are talking about infertility. So what exactly is infertility? So it should be the right definition that we usually talk about, that when there is sexual contact, sexual intercourse or sexual coitus between partners, okay, that too unprotected one. Unprotected means that you are not using any kind of contraceptive method, because obviously contraceptive methods are going to basically try to avoid, contraceptive methods are only used when you are trying to avoid pregnancy. Okay, so if no contraceptive method is being used right now and there is sexual intercourse between partners, unprotected coitus, and that should be for year, 1 year to 3 year, and if still the partners, the couple is not able to conceive a child, then we can say that the couple is infertile. Now obviously the problem can be in any of the partner, the male partner or the female partner. The problem can be with any of the both. Okay, both can also be having problems. So a very good line in your NCERT has been written that in case of infertility in a couple, usually the female partner is blamed. Usually, or we should say almost every time, the female partner is blamed. On the other hand, if we see medical data, medically it is usually found that the male partner had some kind of issues which was causing the infertility issue in the couple. Now that doesn't mean that we should start blaming the male partner as well, right? Blame should not be on any of the person, be it the female partner or the male partner. You should just go for diagnosis and then treatment. Diagnosis means you will be checking out what is the exact issue that the couple is not able to conceive a child. Whether the sperm count is low in the male, whether the sperm motility is low, right? Whether the female is having any kind of issue with the fallopian tube or the uterus, implantation is not able to occur. So all of this will be diagnosis, and treatment options which are going to be termed as assisted reproductive techniques are going to be suggested by the healthcare professional in result of those diagnoses. Okay, so yes, what can be the reasons? If they, this particular line is also very important. If they specifically ask that because infertility is such a huge issue, right, a lot of couples every year suffer from it, what can be the major or common reasons behind infertility in a couple? Okay, so it can be physical issues, congenital issue, diseases, drugs, immunological, or even psychological. I will tell you one by one what all it is. Physical mean when there is some issue with the uterus of the female, with the fallopian tube of the female, with the sperm count of the male. Okay, all of that would be coming in physical. Congenital means something that you have inherited. Okay, some inherited defect or disorder because of which the couple is not able to conceive a child. Disease, for example, we just looked at STD. STD is one symptom, one problem that was caused by STD was also infertility. So because of STD in any of the partner, there can be infertility. Apart from that, if a person is on drugs, so there can be infertility as well. Okay, it can be because of males or females, any of them. Immunological, sometimes what happens that the female's reproductive system can be heightened, having a heightened immune response towards the sperms that might be entering inside the female reproductive tract upon insemination. Insemination is basically transfer of semen inside the female reproductive tract. Now that semen contains millions of sperms. Out of those millions, just a few hundred sperms usually reaches the site of fertilization, which is oocyte. Rest all are going to be basically phagocytosed or killed by the immune system of the female. Sometimes the immune system of the female can be so high that it is killing almost all the sperms, right? Hence no sperms are surviving, hence infertility. Okay, or psychological, when there is some mental issue, when there is some stress, when there is some anxiety, we usually say that okay, it is a mental issue, it is not something physical, right? It should not harm you physically. But the chapter Human Health and Disease talks about how whenever we are stressed, psychologically we are having some issue, mentally we are having some issue, that impacts our nervous system. And our nervous system, if we think about it, has the control of our entire body, right? Nervous system also has control over our endocrine system, and endocrine system was responsible for so many hormones which were responsible for the normal functioning of our body, right? If hormones are not functioning, those hormones also includes our sexual hormones, gonads hormones, testosterone, estrogen, progesterone. So psychological problems can ultimately show symptoms, results in the physical form as well. Okay, so all of these can be reasons of infertility in a couple. Now what should they do? Obviously they should contact a medical professional. They should go for medical consultation. So upon medical consultation, what they will do, the medical professionals, they will first check out what might be the issue, that means the diagnosis, and then the treatment. Okay, so now we will be looking at what exactly are the basically treatment options that we can use. Okay, so what is infertility? For 2 years, 1 to 3 years range you can also take, or the average 2 years also you can take. No pregnancy even after 2 years of unprotected coitus. Unprotected means without any contraceptive method. It will be termed as infertility. Okay, infertility. Okay, we have Zobia as well. Hello Zobia, good evening, how are you? It is your first class as well, I am assuming, right? Am I audible, Zobia?
Yes, ma'am, you're
Great,
audible.
great, great, great. Okay, so I'm assuming. Okay, this is your first class.
Yes.
Okay, okay. So I will give you a little brief of what we are doing. This is basically revision sessions are going on. Revision sessions, we have basically almost, or not almost, we have completed our syllabus once. Now we are completing our syllabus for the second time. We are revising it nicely. But today Jonas is also new in class and you are also new. So I'm going in a little more detailed manner only because with me at least you guys have not finished these particular chapters. Now today we were supposed to do evolution, which we will be doing in a few minutes, right? Before that, in the previous chapter, reproductive health, assisted reproductive techniques or technologies, this particular chapter was remaining. So I was just explaining this. Jonas and I, we both were studying about assisted reproductive technologies till now. Okay, so can you tell me, do you have any idea of the reproductive health chapter? Like, have you ever studied it already in school or have you read it? Because then I will be more careful whether we are revising or studying in
Yes,
detail.
ma'am, I did go through it.
Great.
I did have
Okay,
diagnosis.
okay, great then. So this ART you might be already aware once, right? Assisted reproductive technologies, you are aware?
Assisted, no, ma'am.
Okay, okay. We are doing in detail only. So we'll discuss. Okay, so Jonas,
Ma'am,
till
I was asking if I should watch the videos, like you're saying you completed the syllabus, right? Should I watch those videos or should I start with the revision videos? Are these enough?
revision see, you can also today also you can make the judgment whether it would be enough for you or not, but otherwise we are completing in detail only. So you should be attending the live first. If you face any issues, obviously if you face any doubts, then here I am in live to clear them hand on hand. Videos on the other hand, it is your choice, but I would still suggest to first do it with me, because then I can take doubts, I can see whether everything is clear or not. On the other hand, videos will not be having that kind of assistance, so we should be doing live only. Revision is going to be mostly enough, but if you have issues anytime that it is not enough, can you do a little more detail, or can you please repeat this one, can you basically try to help me with this one more time? So all of this we will do. Okay,
Okay, ma'am, thank you
great,
so much.
great, great, no issues. Okay, so Jonas, till now it was clear, right? So now we will move on to the ART in detail. Can you please tell me once? Great, great, great. Okay, so Zobia, we were discussing what is infertility. So I will repeat one more time. Infertility is basically when a couple is not able to conceive a child even after few years. We usually take 1 to 3 years or 2 years of unprotected coitus. Unprotected coitus means sexual intercourse without uses of any kind of contraceptive method. If contraceptive method is there, that means obviously the couple is not conceiving because of not an infertility, but because of use of contraceptive method, because that is the use of contraception. But if there is no contraception and the couple wants to conceive a child, but still even after 2 years or more they are not able to, then that would be termed as infertility. Infertility can be caused because of any kind of physical, psychological, immunological, any kind of reasons in both the uh, partners in a couple. It can be because of in females or males, any kind of issue with any of the partner can cause infertility. Okay, so what happens if there is infertility? What the couple should do, they should basically consult with a medical professional. They will diagnose, doctors basically they will diagnose that what might be the issue which is causing infertility in that particular couple, and after that there will be a lot of treatment options. Today we'll be discussing all those treatment options. Okay,
Okay, ma'am.
Great, great. So first we will be discussing about test tube baby. So test tube baby is one of the most common ones. We hear about it in a very general manner as well, right? We do not really have to be science students to hear this particular term. It is a very common term. You must have heard it in TV or in some movie, etc. So what exactly is a test tube baby? Are we going to grow a baby in a test tube? Obviously, all of that is not going to happen. But yes, fertilization takes place outside the body in vitro. Okay, two terms which are very important for today's topic are in vivo and in vitro. Can anyone tell me in vitro and in vivo difference? Anyone? In vitro or in vivo? Any idea? Okay, so I will explain. Basically, in vivo means whenever any particular process is taking place or any particular organism is residing in the conditions, in the habitat, it should be naturally. Okay. On the other hand, in vitro would mean that something is happening outside the natural habitat, outside the natural site or the place where it should be occurring. Usually it means under man-made artificial conditions such as lab over here. So if fertilization it is taking place naturally in the fallopian tube, in the ampulla region of the fallopian tube, that will be termed as in vivo fertilization. But if it is taking place inside lab on a slide or in a test tube, that means the fusion of gametes, sperm and ova, is occurring outside in a lab on a slide or in a test tube, then that would be termed as in vitro fertilization. Okay, in vitro means lab, in a lab. It can be on a slide as well. Okay, you must have seen slides in your science lab or a test tube as well. Meroon is here. Hello, good evening, Meroon. How are you? Good evening. How are you? Everything going great? Long time no see, huh? What was happening in your life? So I have chosen to become your mentor. Oh, Meroon, I have chosen to become your mentor. Soon I will be your mentor. I will get your contact details and everything, and soon I'll be contacting you. Okay. So please reply when I contact you. Okay, great, great, great. Okay, so we are continuing ART, assisted reproductive technologies or techniques today. Yes, so test tube baby, it is basically IVF, in vitro fertilization. So now you guys understand all of three that what is exactly test tube baby. Here the baby will not be growing in a test tube. The fertilization will be taking place in a lab, can be on a slide or in a test tube, hence we say test tube baby. Okay, the short for IVF, sorry, in vitro fertilization we use IVF. Okay, so here what is happening, the fertilization and zygote is formed. Now two things can be done with this particular zygote. Obviously, instantly after forming the zygote is not going to stay as a zygote is single cell. I hope all of you are aware of the fact that zygote is a single cell. But we know that even after forming zygote in the fallopian tube, in the ampulla, it was not staying as a zygote before reaching the uterus, right? It had already started the process of cleavage. All of you remember this part? I will... One second. One second, people. Okay. Now look carefully. Okay. From Meroon, I am expecting that, I hope you already know, Zobia, and for Jonas, I'm one more time repeating it. Okay. So everyone listen carefully. So this is, let's say, ampulla. This is ampulla, the site of fertilization. At the site of fertilization, sperm and egg, they met, fertilized and formed zygote. So when this zygote is traveling towards the uterus, it happened in the fallopian tube. When it is traveling towards uterus, right, here it will be implanted in the fundus region of the uterus. It is not going to remain as zygote only, okay? From here only it will start cleavage. Cleavage is basically cellular division without undergoing cytoplasm increasing. Hence the daughters, their cytoplasm keeps decreasing, okay? Cytoplasm in the daughters, for example, here the entire cytoplasm was present in one single cell. Now the entire cytoplasm is shared in two, then in four, then in eight. Now, by the time it reaches uterus, it is already 8 to 16 celled stage, which is termed as morula. Everyone knows about morula? By the time it reaches uterus, it is already morula. Any idea about morula?
Yes, ma'am. I
Very
read about early morula is 8 and 16.
Yes, very good, very good, very good. So by the time it reaches uterus, it should be already 8 to 16 cell stage morula, and then in the uterus it will be converted into blastula or blastocyst, and this blastula is going to do what? This blastula will get implanted, okay? Now the thing is, after forming zygote in the lab conditions, two things can happen. The cleavage will start instantly. If the cleavage has started and 8 to 16 cell stage has reached already, then there is no meaning of transferring from externally. Doctors, with the help of a syringe or a long pipette like structure, they will transfer the formed cell mass, a little cell mass. We cannot call it zygote. We will call it zygote only and only it is if it is before the 8 to 16 cell stage, okay? If it is, then we will transfer it in the fallopian tube. If the 8 to 16 cell stage has not reached, we are going to transfer it into fallopian tube. From there onwards, it will take on its natural process. How naturally? If naturally fertilization would have taken place, then how it would have been implanted and everything. But if 8 to 16 cell stage has already been reached, we cannot transfer it into fallopian tube, because naturally 8 to 16 cell stage should not be in fallopian tube. It should be already in uterus. So then it will be transferred into the uterus directly, okay? So whenever we are transferring the zygote up to 8 blastomere. What is blastomere? Then if this is basically 4 cell stage, then one single subunit of the particular cell mass is going to be termed as blastomere. So if it is at up to 8 blastomere, only and only 8 blastomere has been reached, then we are going to call it zygote intrafallopian transfer, okay? We are going, it is not really zygote if 8 divisions has, if we have reached 8 cells, that means at least 3 times the division has already occurred. So it is not really zygote, but still we are transferring it into the fallopian tube, okay? Zygote intrafallopian transfer, ZIFT. But if more than 8 blastomeres are there in that particular cell mass already, then that will be termed as IUT, intrauterine transfer, okay? ZIFT is zygote intrafallopian transfer, the transfer of up to 8 blastomere stage in the fallopian tube. Intrauterine transfer is IUT. More than 8 blastomere are there, hence we are transferring it into directly uterus. From there it will get naturally implanted and pregnancy will occur. Clear? Any doubts in ZIFT or IUT till now? Any confusions? Any doubts? Okay, great. So everything clear? Okay. Clear? Everything? So if we talk about test tube baby, it is going to have two processes. One is IVF. Great. One is IVF, intra vitro fertilization, and second is embryo transfer. Embryo transfer can be done by two methods, ZIFT or IUT. Okay, that was the first technique. Test tube baby. Now, why do we need to do test tube baby, ma'am? In which couples can we use the test tube baby method? Okay, so in the couples which are having issues in fertilization, the fallopian tube is normal, okay, the uterus of the female is normal. The only issue was that the gametes, they are not able to get fertilized, they are not able to meet. Okay, if and only they will meet, they will be present at the site of fertilization at the same time, then only they will fertilize, no? So if there is sperm count too low, and obviously the female's system eats up so many sperms, the immune system attacks the sperms, so all the sperms are basically getting killed before reaching ampulla only. In that case, if sperm count is low, obviously infertility will occur. Hence we can use this particular method, because we are selecting that yes, this sperm, nobody can kill it now, because it is in lab condition, there is no immunity of the female right now, and it should fuse. So it fuses basically, because that is how the chemistry of the egg and sperm is. Okay, they have the chemical receptors and everything which are supposed to be making them fuse with each other. So if sperm count is low, there is some issue with the or any kind of issue which is inhibiting fertilization, then we can use test tube baby. Okay, so this is the first part. Now what if, ma'am, the mother is not having that, basically the female is not having any kind of normal conditions in the fallopian tube or uterus? That means the mother had a normal egg and the father also had normal sperms, but the mother is having issue in fallopian tube. Because of that, what cannot happen, we cannot transfer the we cannot perform the zift, zygote intrafallopian transfer. Then we will perform the IUT, intrauterine transfer. But if there is some issue with uterus also, then the pregnancy in the female, in that particular female, cannot occur at all, right? The growth of the baby in the womb cannot occur at all, because the womb is basically uterus, it is having issues. Okay, there is some abnormality in the uterus. In that case, surrogate mother is required. Okay, so now you understand why surrogate mother would be required, because the mother, the female from whom we are taking the egg biologically, who is going to be the biological mother of the baby that is going to be produced over here, she was having some problems with the uterus. Now uterus is supposed to be the site of the entire growth of the baby for the next 9 months, so that cannot occur over there. So we will choose a surrogate mother with a normal uterus. Okay, and we can transfer the ready-made embryo. No, we will not call it embryo, we will call embryo only after implantation. We will transfer the zygote or the cell mass directly. We can perform zift also. We can, that means a transfer in the fallopian tube of the surrogate. Or secondly, we can perform IUT, intrauterine transfer as well. That means in surrogates uterus directly we can transfer that cell mass that we have produced in lab condition. So biologically, who is going to be the mother? This particular female is going to be the problem. Sorry, this particular female is going to be the parent. Okay, she had uterus problem, so she could not basically carry the baby for the 9 months. So we had to use a surrogate mother. So from the biological parents, biological mother and father, sperm and ova is taken. In vitro fertilization is done up to 8 blastomere. If it is, then zift. If more than 8, then IUT, and that is done in some other female. Now this female is going to carry the baby for next 9 months, but she is not the biological mother. Genetically, she doesn't have any kind of relation with the baby that is growing inside her womb. Okay, so that is the surrogate mother concept. Clear? Surrogate mother concept clear to you people? If any issues, you can tell me. Yes, Zobia? Okay. Zobia, your voice got a little disturbed. So can you repeat, please? You had any doubt, or is it clear?
No, ma'am, I said it's clear.
Great then. Okay, let's move forward. Now, third is gift. Now, you guys must be thinking, my God, what is ma'am? What is gift? What is zift? Zift is zygote intrafallopian transfer. Gift, I will tell you a easy trick, right? Previously, the biological mother, right, the couple who wants to conceive a child, she was having normal gamete, normal egg was there. Problem was with the uterus or with fertilization. Hence, if fertilization is the issue, we will fertilize in lab and we will transfer the prepared zygote in the biological mother only. If problem with uterus, we will fertilize in the lab and we will transfer the zygote in some surrogate mother. But what if the problem is with the egg only, right? That means the ova in the mother, the biological mother, the couple who wants to conceive child, the ova is not at all viable. The gamete that is produced by the mother, it is not viable. It cannot produce a healthy baby. Okay, she can never get pregnant by her own ova. So what can we use in that case? Then a very easy trick to remember is that she can get the gift of ova, the gift of egg from some other person, and that person is going to be a donor. Okay, from a donor, the female can get a egg. Now that egg can be directly transferred to the ampullar region of the couple who wants to conceive. Okay, the egg is from some other person and it is transferred into the ampulla, fallopian tube of some other person. So biologically, obviously, the child is not going to be hers, right? But she is going to carry it for the next 9 months. Okay, she is going to be the official mother of the baby if donor is involved. Okay, donor's only job was here to donate egg. Okay, the egg was transferred directly into the fallopian tube of the mother. Fallopian tube was normal, uterus was also normal, so she could carry a baby, but ova was not functional, ova was not viable, so she had to take the female gamete from some other donor. So biologically the baby is going to be of the donor and the male partner of this particular female, but obviously officially it is termed as that she only is the mother. Okay, it can be done in case of males as well. In case of male donor, in a couple, the female might be completely normal, but the male partner might be having issues with sperm count and everything, and the issue is so huge that it cannot be resolved with any method. Okay, you cannot resolve it by IVF or anything. The sperms are completely non-viable. So in that particular scenario, sperms from some male donor will be taken, and those sperms plus ova from this mother if she is normal, and then zygote will be produced. Okay, and these sperms we will understand later on. It can be transferred by artificial insemination. Artificial insemination I will tell you later on. So right now you do not have to get in details of this. Artificial insemination means ejaculation from the donor will be getting done. Okay, and from there, ejaculation means, I hope you guys remember, I have already told you, ejaculation means the releasing of semen outside the body, not inside the female reproductive tract. Okay, so outside we have got an ejaculation done, and now artificially with the help of a pipette or with the help of an injection, we will transfer the semen inside the female's reproductive tract. Okay, so that can also be done. So gift is basically gamete intra-fallopian transfer. We are transferring a gamete into the fallopian tube of the female, and the gamete is of a donor. Hence, biologically, the baby is of the donor, but obviously, otherwise, the mother is the person who will be carrying the baby. Okay? Clear? Gift is clear? Yes. Okay, okay. There is a huge issue. No, parent dispute will never be there, Maroon. Whenever any person, okay, in case of surrogacy as well, there is a huge paperwork and a huge legal paperwork. Okay, I will tell you in both the cases. In surrogacy as well, a huge paperwork is being done between the surrogate mother and the couple. Okay, and it clearly mentions that the mother, surrogate mother, is only carrying the baby. She will be never having any kind of official right over the baby. Okay, she's just carrying, and after giving birth she has to hand the baby over to the parents. In case of donor, the donor gets paid. Okay, the donor gets paid to donate their sperms or egg and ova, and when they are donating, they also have to sign a huge legal document where they make sure that now they would not be having any kind of right on that particular baby, even if the biological parent is that particular donor only, but the official parents will be considered the one who has taken the donated gamete. Okay, so they have to sign legalities are there. So they do not have any kind of right over the baby. Obviously, no couple would do that, right? Or if you are growing up, if you are upbringing a child and everything, you are emotionally attached to a child, and someday some other person comes and they say, oh my god, this is biologically my baby, you have to give it back to me. So it is going to be very much huge issue, ethically as well, officially as well, legally as well. So a legal thing is being placed in such scenarios. Okay, so you do not have to really worry about that later on. Okay, next we have ICSI, intracytoplasmic sperm injection. Okay, so here I hope you guys remember the main majority of the times they ask in this particular technique which type of fertilization was done, in vivo or in vitro. So in test tube baby it was in vitro fertilization. Here can you tell me what type of fertilization it will be, in vivo or in vitro? In vitro fertilization or in vivo fertilization? In vivo, very good. Because we are just taking only artificial thing we have done over here. Very good, Jonas. Only artificial thing that we have done is we are taking gamete from some other person, but we are transferring that gamete into the fallopian tube so that natural fertilization can take place. In the fallopian tube, naturally fertilization is supposed to be taken place. Okay, next is intracytoplasmic sperm injection, and this one we have studied in biotechnology as well, right? Something related to this we studied in biotechnology as well when we were transferring plasmids in the animal cells. So it was not sperm injection. Very good, very good. Not gene gun, not gene gun. Gene gun, gene gun or biolistics gun or gene gun was used in plant case, plant cells. Cell injection method was there. Okay, so here what we do, we will take, yes, in animal cell injection was there. So what we will do, we will take a slide, okay, and you can watch. This is such a beautiful thing to watch on YouTube or any of your social media. You might find some videos because under microscope they are doing all of that, and they usually take videos of all of that in microscope. Okay, so you can see this particular process. There must be a YouTube video, I have watched it, so you can search ICSI, okay, and a video will be there. There you will see that on the slide they have a slide like this, and then they have a small, very small tools are there. Obviously they have a small tool holding the ova in place. This tool is holding the ova in place, and then this is the nucleus of the ova. Then they bring the sperm injection. Okay, injection is there, very tiny needle, obviously, because the injection needle has to pierce through the cytoplasm and transfer the sperm without harming the egg, right? So there is injection now, and in this injection what they have done, there is presence of sperms inside the injection. Okay, one sperm only we have to transfer, so sperm is there. Next thing what will be done, they will inject the sperm inside the cytoplasm of the egg. Okay, so this is intracytoplasmic. They are sending the sperm inside the cytoplasm of the egg. This is egg and this is cytoplasm of the egg. And this is the sperm. Now why it is done? Obviously, again, in the case of when the male is having some issue with the motility in sperm. Okay, if sperm is not motile, then what we can do, we cannot rely on natural fertilization. We cannot even rely on the normal in vivo fertilization where we just left the sperm and the egg in a test tube and the sperm automatically fused with the egg. Here motility in the sperm is not at all present. Hence what we can do, we can bring the sperm directly to the cytoplasm of the egg and we can transfer it there inside, and once transfer is done, obviously the nucleus they will fuse and zygote will be created. Okay, so that is intracytoplasmic sperm injection. By injecting sperm inside the cytoplasm of the ovum, we are making the process of fertilization possible. So can you tell me whether it is in vivo or in vitro fertilization? If we are doing such heavy medical work, whether it is in vivo or in vitro fertilization? Something happened with my throat today. In vivo. So you so you think all of this we are doing in mother's womb, in the fallopian tube of the mother? Have we put a injection and then a slide and then a holder of the egg all inside the fallopian tube of the mother? Then should be in vitro. Yes. Jonas, it is clear, Zobia? Why it should be in vitro? Yes, very good. Zobia, it is clear? What happened? Zobia went to grab a snack. Okay, let's move forward. Okay, let's see. Next is AI, artificial insemination. Okay, this is the artificial insemination I was talking about in case of donor male. Okay, so here what can be done? The sperms can be having some kind of issues. Okay, or the male can be having completely normal sperm count and everything, but there can be a issue with erectile dysfunction. What is erectile dysfunction? I will tell you. Erectile dysfunction is issue with erection of penis in male. So in that case, what happens? I hope all of you remember that we had presence of spongy tissues in penis, and those spongy tissues were making sure that upon sexual arousal, the penis, those tissues, they basically trap a lot of blood inside the penis, and because of that blood inside the penis was getting erected, and obviously it has to enter the vaginal opening, the female's reproductive tract it has to enter, and so to enter it has to be an erectile structure. If it is not an erect structure, it cannot enter the female's reproductive tract at all. Okay, so for that it has to be erected. But if there is some issue with the male which is causing erectile dysfunction, then what we can do, right? Then we will have to ejaculate the semen outside only. Release of semen outside the body is ejaculation, that only we will have to perform. Okay, now that semen that we have collected medically, we will inseminate it artificially. Artificially means with the help of a pipette or injection, we will send it directly into the female's reproductive tract. Okay, so it can be used in case of erectile dysfunction or when the sperm count is too low. Sperm count is too low means obviously we know 60% of sperm from millions of sperms that are produced should be of normal shape and size, and out of which 40% should show vigorous motility. If that is not present in any male, then we say that the male can be having fertility issues. Okay, so if you remember this, we studied in human reproduction that all those millions of sperms produced by a male, if 60% are of normal shape and size and 40% are showing high vigorous motility, then only out of those millions at least one will be able to reach the ampulla and fertilize the egg. If that is not followed up, then male will be having fertility issues and we will have to perform artificial insemination. Artificial insemination, that means we are directly injecting the semen into the fallopian tube of the female. Okay, we are direct transfer of semen in fallopian tube of female by injection or pipette. Basically tube like structure, very thin. Okay, that is artificial insemination. Artificial insemination clear to everyone? What happened? What is happening? Such a white slide I've never seen in my life. Okay, great. Now we start the evolution part. Such a white slide we had. Oh my God, in my entire life, in my entire teaching career, I have never used such a white slide. I don't know what is happening with the board today. Okay, so obviously the reproductive health took more than expected. It's just, yes, it is just transfer of semen. Okay, semen got withdrawn outside the female's body. Okay, in a natural coitus, what happens? The penis, it enters inside the female's reproductive system in the vaginal opening. and then their transfer happens naturally. Okay, if that is not able to occur because of erectile dysfunction, or it is able to occur, but still the couple is not getting pregnant because the sperm count is too low. So when it is getting ejected in the female's vaginal opening, vaginal canal, then it has to go through the cervix, then it has to pass through uterus to reach the ampulla, right? In this whole time, there are so many immune cells which are going to eat up sperms. Okay, sperm is healthy in everything, but the sperm count is low. Hence, enough sperms are not reaching the site of fertilization to fertilize the egg, or the semen is not able to get inseminated into the female's tract because erectile dysfunction is there. Okay, penis is not getting erected only, so it cannot enter vagina. In those cases, just semen can be taken out and transferred into the female's fallopian tube directly with the help of a thin pipe. Okay, simple. Okay, great. Now we will start evolution. I think I should have just made it, oh, it's just 8:30. Half hour is left, we will do it, no worries. Okay, so evolution we will do now. Introduction we will start. Okay, so what exactly is evolution? Evolution is when life came to earth, it was extremely simple. Okay, and now look at us, we have digital boards, we have cameras. I am teaching from India and it is reaching directly to you people, right? And you are sitting on in some other part of the world. So all of this is basically development, and all of this was possible because of evolution. Okay, so changes in life form is basically evolution. We need proof of existence, non-existence. Okay. We will check all of that. So study of evolution is what? Study of evolution is evolutionary biology, the complexity of life. Yes, this evolution is going to make you think about life so much now. You will be, my God, that this is how we came, ma'am. You would be thinking all of that. Evolution is full on theory. Reproductive health was also full on theory. It was almost social science, apart from the STDs or the art. Rest everything was social studies only. And similar forms, in similar manner, evolution is also a lot of philosophy of life. Yes, so we are going to study philosophy of life today. Okay, so what exactly is evolution? Changes in life form. From the simple cell we were formed, that is evolution. Okay, and when we study evolution, right now what we are doing? Right now we are doing the study of evolution, that is termed as evolutionary biology. Okay, simple. Now let's understand what exactly happens. So the very first paragraph of this chapter is actually very beautiful. Okay, so your NCERT is so well written that there are just so many like deep very philosophical lines in your NCERT. I will bring a few. I have seen on social media. I don't know whether you guys follow the social media content related to your NEET exams or not, but a lot of students, they post such beautiful lines from NCERT which are extremely philosophical. And one such line we have in our book over here. Okay, so they say that whenever you're looking in the sky, you are looking at past. So can you guys tell me how exactly are we looking in the past whenever we are looking at the sky? Anyone? Light years difference. Very good. Very good. Stars are in the past. Very good, Mehroon. So Mehroon knows all the philosophy. So people, I hope you are aware that when light reaches to us, right? I am able to look at this particular pen right now because light got reflected from this pen. It entered my eye, and then my retina, all those cells present on my retina, neurons, they send the information to my brain. Okay, so it was light reflected from this particular pen. When it reached my eyes, then only the pen was visible to me. NCERT is yes, that's why I read the NCERT with you guys, na? Whenever I have time. Yes. Yes. So whenever you're looking at the sky, that means you are able to see the stars because the light reflected from those stars, from those planets, is entering your eyes. And for that light to reach your eyes, it took many years, because lights are present at thousands of light year difference to us. What exactly is a light year, ma'am? Light year is that this beautiful galaxy of ours, Milky Way. Right now we are somewhere at some particular point in that Milky Way. Okay, so here we are such a tiny dot that this dot that I have made, it must be like so huge. It is even bigger than a solar system. Okay, so this is our beautiful Milky Way, and then we have another star in our Milky Way somewhere. Okay, so let me draw a little more distance, because then only it will be easy for you to check out. Okay, so if we check out the difference from the star to reach to us, it took many years. Okay, and for the light to reach, time taken by light to travel between 2 points in the space is termed as a light year. Okay, light year. How much distance light covered? So we are 1 minute-ish behind in that's present. 1 minute-ish? No, it doesn't. Light travels very fast. You guys must know about the speed of light, right? Not 1 minute-ish. How are we 1 minute behind? Not 1 minute. If you are looking at the pen, oh, out, no. Imagine you are right now moving the hand and you're able to see the hand movement. It doesn't really take a time. Okay, so what exactly is light year? Time taken by light to travel in 1 year. Okay, in 1 year. Light years is, sorry, distance traveled by light in 1 year. Not time taken. I will write it over here, don't worry. Distance traveled by light in 1 year is a light year. Okay, so it takes many years for the light from those stars to reach to us. Hence we say, for if we travel to that object, we'll see fast forward in real time. Oh my god. So Maroon is a fan of science fiction. You are a fan of, huh? You guys have watched science fiction and everything. So now we will understand. So life, if we are discussing the change of life, evolution is basically changes in life. So to understand changes in life, we first have to understand how life came, right? And for us to understand about life, we have to understand how Earth came. And to understand how Earth came, we have to understand how universe originated. Okay, so we will be looking at the origin of universe, origin of Earth, origin of life, etc. So when exactly origin of universe occurred? Okay, there is a theory of origin of universe as well, Big Bang theory. We will be studying about that also a little. But if you are very much interested in science fiction and everything, then you should, it would not be science fiction. This is pure research, obviously. It is a theory, because obviously there is no proof of all of that. We were absolutely not present at the time of origin of universe, hence we cannot prove it. Hence we say all of that is theory. Theory is basically a concept that was given, but there is absolute no proof of that. Okay, so origin of universe was 20 billion year ago. BYA, Baya is a billion year ago. MYA, Maya is millions years ago. Okay, so 20 billion year ago, universe originated by a Big Bang theory. We will understand about that. After that, just 4.5 billion year ago, Earth came into picture. Okay, Earth formed. We will understand how Earth formed. Then after that, 4 billion year ago, that means after 500 million, after 500 million years, 500 million years after the Earth had formed, life started. Origin of life happened. And obviously that life was not as complex as we are. It was extremely simple. Okay, it was in the form of non-cellular life. So non-cellular life came when? Non-cellular life came 3 billion years ago. Okay, here origin of life, when it occurred, it was in the form of molecules only. Very simple molecule. From those molecules, when those molecules, they interacted with each other, then non-cellular life came into picture. Zobia, you were having any technical issues? Any technical issues you are having, Zobia? Okay, it is still not, I think, working. Zobia, am I audible to you now? Some technical issue you are facing? Okay, not audible, I think. We'll see. Okay. So here when we talk about origin of life, it is a very simple in the form of molecules. When those molecules interacted with each other, they formed some first non-cellular life form. That means it is still not life, but it is in the form of proteins, amino acids, etc. Now when those proteins and amino acids, they interacted with each other, they arranged themselves as a cell, then cellular life came into the picture. Okay, so 20 billion year ago, universe. After that, just 4.5 billion year ago, earth. 500 million years after the earth, that means 4 billion year ago, what came into picture? Our our life, origin of life in the very simple form, such as ammonia, water, etc. Okay, after that, how many years ago? 3 billion year ago, non-cellular life came into picture. That means proteins, etc. Okay, prebiotic life, it is termed as RNA, proteins, carbohydrate. When they interacted with each other 2 billion year ago, cellular life came into picture. Okay, cellular life, when it came, it was obviously unicellular life. So after that, it got converted into multicellular life 1.5 billion year ago. So that was how the transition was. Okay, so much of time it took. Unity is the key. Yes. Yes. So it is basically a random arrangement of the cells. Why though? Why? Because see, one one thing in science that you study is that the molecule, the particles, they are in constant motion, right? They are constantly vibrating, right? So when those vibrations occur, the particles, they move here and there, and so much of years means unlimited, right? Infinite probabilities of them arranging themselves into different arrangements, and different arrangements led to the right arrangements as well. Right arrangement means, if we talk about water, obviously we had, when all of them are interacted with each other at the right arrangement, right bonds got formed and water came into picture. Okay, okay, so hydrogen and all of these elements. Yes, yes, so they kept interacting with each other, hence it took so many years. No, 20 billion year ago, where what happened, universe originated, and it was just 1.5 billion year ago when actual multicellular life originated for the first time. So obviously so many infinite possibilities of arrangement of molecules were there, and in India we had, sorry, in our earth we had the perfect right conditions, hence life was possible. Okay, otherwise life would have not been possible. So it was the right arrangement of molecules that life came into picture. Okay, so the not in India, universe, that was a tongue slip. Earth. Okay, earth is the perfect arrangement. Okay, perfect arrangement of molecules, and this perfect arrangement of molecules might have been occurring somewhere else in universe. Do you guys believe in that? This particular perfect arrangement that we have over here on our earth, it must have in some other part of our universe as well, right? Obviously universe is so huge, somewhere else also it might have occurred. Hence we say that it is not completely impossible that somewhere else life also exists. Okay, so now we will be understanding the origin of universe. Okay, origin of universe. So it is explained by Big Bang theory, and your NCERT again has done a little wordplay over here. NCERT has not written that Big Bang theory explains origin of universe. NCERT has written Big Bang theory tries to explain origin of universe, because explanation of origin of universe is definitely not there, right? We can only and only try to find the right explanation. To have the exact explanation, we should have been present at that time, and there is no way that we could have been present at that time. Yes, yes, Zobia, evolution had just started. So I was asking, were you having any technical issues? It has just started. It's kept inaccessible because we didn't solve the grid and pollution problem. Yes, yes, Meroon. All of that is also there. Zobia, evolution has just started. Okay. We had just looked at the first introduction paragraph. Okay, great then. So Big Bang Theory, please, every time you should mention attempts or tries to explain the origin of universe. 20 billion year ago, there was a huge explosion, and that explosion was so huge that it is completely unimaginable. We cannot describe it in the physical terms. Okay, explosion which cannot be explained in physical terms. Physical terms means whenever we talk about an explosion, for example, we usually say the Hiroshima Nagasaki explosion was very huge, right? It had so much of destruction. So that particular explosion which almost erased two cities, right? It was so huge. Still we can still attempt to explain it in physical terms, that it was such a huge explosion, it erased two cities. But Big Bang Theory was such a huge explosion that we cannot even think about explaining it in the physical terms. It was extremely huge. Okay, hence obviously universe came into picture. How it actually happened? So there was a mass of high energy particles, and that high energy particle mass was basically constantly vibrating and vibrating, and then ultimately the energy was too much to be consumed by this particular mass, so it had to burst, and that bursting of that particular mass led to formation of universe. So what exactly was present in that mass? Obviously a lot of energy. Apart from that, dust particle elements such as hydrogen, carbon, oxygen, all of this was present in that particular particle of high energy which exploded. Okay, so particles, now obviously after explosion, when you have a small particle and it explodes, so all of those particles, they are going to disperse in a huge space, and hence universe basically had expansion of particles in all over different directions, and those particles slowly they started cooling down. Okay, after explosion obviously everything is very hot, everything is with high energy, so slowly they started cooling down. So when things cool down, they obviously again try to basically mix with each other, form clumps, form groups. Okay, so when particles they started forming together, groups and clumps, they started forming stars, they started forming planets, etc. So that is how also our earth came into picture. Temperature came down, helium and hydrogen formed later on. Okay, helium and hydrogen also formed later on. This particular line is clearly written in your NCERT, that after explosion the temperature was coming down, helium and hydrogen also formed. After that, what happened? This gas cloud, this huge gas cloud, it started basically coming together and forming our earth. So gas cloud, or we also call it cosmic dust. Cosmic dust has basically dust particles plus gases. It was coming together to form nebula. Nebula is what? Dust, gases. Apart from that, it is also having different elements. Okay, stars. So that is a nebula. Nebula is a dust plus gases plus stars. That is a nebula. Okay, so this made the galaxy. When all of this aggregated, this formed huge chunks, groups. Then galaxy came into picture. Galaxy formed because of that. Okay, so that is how galaxies formed. Now we talk about Earth. Earth formed by those mass, those dust and gas clouds when they came together. Okay, after condensation of all of those gases that are present in galaxy, Earth was formed 4.5 billion years ago. At that time on our Earth, everything was super hot, high temperature. Okay, 800 degrees Celsius of temperature. Yes. Yes, that happens. And dead stars. Stars also have a expiry date. They obviously die. Okay. Our star, our sun. Yes, we are stars. We can say that. Our sun is also having expiry date. So sun is also going to collapse sometime. Okay. First it is going to burst, and then it is going to become a dwarf. Dwarf is basically a very tiny star not having much of light. Okay, so all of that will happen. So but all of this is very far. Okay, you guys will not even be there to see all of that happening, so don't worry. Okay, so when all of this was happening, 4 or 5 main elements that were present which started life. Okay, so here we have our methane. Methane was present in our environment. Apart from that, ammonia, methane, ammonia. Apart from that, we have our carbon dioxide and water in the form of water vapor. these 4 particular elements were present. Okay, they constantly started reacting with each other. How exactly? Now, during this time, our Earth is not having any environment. Environment means right now we have a very thick layer, a blanket of ozone on our head, which is protecting us from so much, right? Which is protecting us from UV rays, harmful UV rays of sun. Otherwise, we couldn't survive. So, ozone has not formed yet. It is going to form. We will talk about that. So, if ozone is not there, that means UV rays are directly falling on all of these elements. And when UV rays falls, I hope you know that UV rays are very much, basically they will cause a lot of reactions to occur. So here what happened, upon UV exposure, water broke down. Water was getting broken down into H2 and O2. Okay, hydrogen, what was happening with hydrogen? Hydrogen was a very light. Hydrogen is a very light gas. So it escaped the environment. On the other hand, oxygen is slightly heavy gas. So oxygen combined with other elements. Which elements? All of these elements, and started forming new different elements. Okay, so that is how free oxygen came into picture. And that is also how free oxygen also made ozone later on. We will talk about ozone some other time. Okay, so due to high temperature, all of this was happening. Now the thing is, because here the entire atmosphere is very much reducing temperature. Reducing atmosphere means that free oxygen cannot exist. Okay, the oxygen will, upon forming, oxygen forms and it will quickly combine with some other particle, with some other element, because there is just so much of free hydrogen present. So no free oxygen is available. It is quickly combining with some other element and forming something else. That is termed as a reducing atmosphere. So in the beginning on our Earth, the atmosphere was reducing. Reducing means free oxygen is not able to survive. It will quickly combine with some other element. Hydrogen and everything is there. Okay. Temperature was very high. About 800 degrees Celsius of temperature was there. Okay. Now, next up, what happened? Okay. I think I do not have a slide over here. Wait, I will explain. No worries. Okay, no worries. We will look at this. So when Earth formed, obviously it had huge bumps. Okay, Earth is not smooth. Okay, very huge. Whenever we look at our moon, we say those spots are present. Those spots are huge craters. Craters are basically very deep. What word we would use? Very deep. Craters is what? Like deep embedding into the surface. The surface is not round and smooth. The surface is like this. So if the ozone is the reason why biomolecules are limited to what is today, and we are mocking yes, see ozone, when we say that sun exposure is causing cancer, all of you have heard about that. Yes, craters, craters I know. All of you know about that sun exposure causes cancer. How is it causing cancer? Because UV rays are very much mutating. Okay, it is mutagenic, which means when it is coming in contact with your DNA, when it is falling on your skin, it is coming in contact with your DNA, it is mutating your DNA. Okay, so UV rays can cause DNA mutations. So hence cancer can be caused by that. So yes, UV rays are mutating in nature. By making hole, we are harming ourselves. Yes, that is definitely happening. So what is happening over here? So as the temperature will come down, all the water vapors that are present in the environment, okay, a lot of water vapors are present in the environment. Those water vapors, they will start falling on Earth as rain. Okay, as rain. So as the temperature is cooling down, a rain on our Earth started, and then that particular rain lasted for many years. Okay, so it rained for millions and millions of years on our Earth, and hence we have oceans today. The ocean and sea that we see today, it is as a result of years and years, millions of years of rain of that particular water vapor that was earlier present in our atmosphere, and that rain fell into these craters and it filled all those craters, forming our oceans and seas. Okay, now when all of this was happening, before the rain, what was happening? A lot of elements. Which elements, ma'am, to be specific? These elements, they were combining with each other. Combining how exactly? Okay, one more element is left. Okay, CO2. We will write CO2 only. All of these were combining with each other and they were forming our amino acids. Okay, nucleotides, simple sugars. You guys must have read biomolecules already. Trenches, I don't know what you would call those huge uh, craters. Okay, so you must have read biomolecules already. Biomolecules means that we are made up of these elements only, right? Hydrogen, nitrogen, carbon, oxygen, all of this is making us, right? We are made up of all of this only, but all of this had to react in a certain manner to form amino acids, nucleotides and simple sugars, and that happened. How? Because temperature was high, 800 degrees Celsius. There was a lot of thunderstorms, lightning, and lightning was providing energy for the chemical reactions to take place, the breaking and making of bond to take place, so that from these elements, amino acid, nucleotides, simple sugars can be formed. Okay, and this particular origin of life, which is termed as chemical or evolution or chemical origin of life, is explained by O'Brien and Haldane theory. We will read about that as well. Okay, so right now you need to understand that rain happened and all of this, whatever reaction was happening, it got transferred into oceans as hot soup, hot or life soup. That means a very high temperature water is filled in oceans and all of these elements are present which are constantly undergoing reactions to form all of this. Okay, so if they ask about life soup or hot soup, you will know what is life soup or hot soup. Yes, we are soup. We came from soup and that is life soup. Great, great, great. Now we will understand theories of origin of life. Now obviously we were, when we were so not so advanced in our research and science, we were not able to tell that we came from chemicals. Obviously different type of theories were there to explain life, how life came into picture, and all of those theories are given over here. Okay, and they are termed as origin of life theories. First one is panspermia. So Greeks used to believe in this particular theory, and Greeks used to say that life came from outer space and it came in the form of spores. Spores, I hope you know that right now in our atmosphere we have a lot of fungi and microbe spores. Yes, very good, very good. So fungi and all of these, for example yeast also, they reproduce with the help of spore. Fruiting bodies form in them and those fruiting bodies, they burst. So spores, which are basically seeds of those microbes, they burst into our environment, and now even if we pack some food in a very close container, after few days when we open it, there will be formation of mold on it because spores transferred into that because it was present already in our air. So similar way, Greeks used to say that we also came from spores, and those spores, they came from outer space. So this is what panspermia is. Richer was the person who was leading this theory. Okay, Greeks used to believe in this theory, and Richer is the person who was leading this theory, like who gave this particular theory. But you do not have to remember Richer. You just have to remember what was panspermia, that life came from outer world in the form of spores. So you guys were spores. Yes, the Richer name is back. Okay. Now, next up, we have spontaneous generation or abiogenesis. Spontaneous generation means the origin of life was very spontaneous or from non-living particles. Later on, when we study chemical evolution, we say that yes, it is from chemicals only. Chemicals are non-living particles only, but still, this particular theory was a little very much absurd, I must say. Okay, so this was given by Aristotle and Van Helmont. Aristotle is a very famous scientist. I hope all of you know his name. He has done a lot of different works in our science. Okay, this particular work was also given by him. So Aristotle, what he noticed that he had wheat straw. You know, wheat straw is basically stem of wheat. Yes, he has given a lot of work. Whenever we study bio, we see a lot of time Aristotle being mentioned. Okay, okay. I got the cut the class, ma'am. So your class time is over and you guys didn't tell me. I was also going in flow. So this we will continue. Okay. Evolution is left. We will continue. Homework. My God. I was expecting too much and today I have given you a DPP. But DPP you won't be able to solve fully because complete revision is not done. We will do it. Okay. No worries. We will cover it. Okay, great. Thank you so much, guys. Thank you so much. I will see you in the next class. We will continue evolution. And in about 2 sessions, the entire revision of evolution should be done. Okay? Okay. Thank you, Jonas. Thank you, Zobia. Thank you, guys. Thank you so much, Mehrun. I will see you in the next class. Till then, bye-bye. Take care. Have a lovely day. And yes, join on time next time. Bye-bye. Thank you so much, guys. Bye-bye.